My name is Juan Espinosa, Director of Student Success here at the UCLA School of Nursing, and today we're very excited to kick off the Faculty Spotlight program with Dr. Nalo Hamilton. Go around for some introductions. Dr. Nalo Hamilton is an Associate Professor with the UCLA School of Nursing and the Associate Dean of Equity, Diversity, and Inclusion. She has experience with curriculum development and the education of the Advanced Practice Registered Nurse, APRN. Her unique background in biochemistry, molecular biology, and nursing, coupled with her clinical expertise, gives her a unique perspective on education and the sciences of clinical development. Alrighty, Dr. Hamilton, take it away.
Good afternoon. Okay, I'm there, and I think some of you see me. I look very far away, like a little raisinette, and that's perfectly fine. But I'm going to take this opportunity to talk a little bit about myself as the Associate Dean of Equity, Diversity, and Inclusion. Should have changed that. And also, then I will segue into my research. So right here, how I look at diversity, equity, and inclusion, we have here at UCLA, it originally started, this position, this office, both campus-wide, started with the Moreno Recommendation or Implementation Committee. And they wanted UCLA to take a look at diversity, equity, and inclusion across campus and in all units. At the time, though UCLA is located in a very metropolitan area, we weren't being very inclusive. And so it started with a vice chancellor's position for EDI, and it has moved to having equity advisors in all of the units through the school. When it comes to nursing, we're a little bit along the same vein. As you can see, I have several publications that have dealt with the retention of faculty of color in academic nursing, as well as dealing with how patients that are non-white actually have mistrust with their providers, and also with the recruitment and retention of diverse students in the nursing academy, and specifically in women's health. What's very interesting about nursing, as much as we give care, as much as we are the #1 trusted profession, we ourselves are not as diverse as we need to be. A lot of folks are hearing about the faculty shortage, so I'm going to take this opportunity to make a shameless plug. Is that the reason why we have a faculty shortage or a nursing shortage is because we don't have enough nursing faculty. And what would benefit us is to have more students who are in our BS program to consider a PhD program, our students who are in the APRN program to consider getting a PhD, as well as looking at getting a doctorate of nursing practice. The reason why is these two professorates are necessary to maintain the nursing profession. So when you have enough faculty, then you can admit more students. And therefore, we are able to address the nursing shortage on both ends. We also have to remember that many times our most diverse population of students in nursing is actually at the bachelor's level. So our pre-licensure students are the most diverse. And as we move into the other layers of the academy, such as our DNPs and PhDs, we lose that strata. And we know that we are up against something called money. You get paid a lot more working clinically than you do get paid in academia. But there are perks of academia that you will never get when you work in a hospital or in an ambulatory care. And if you'd like to discuss those further, I am happy, thrilled and ecstatic to chat about with you. Now, many times in this position as a faculty, there's a lot of things that go on. And I always use this highway because many of us are very familiar with the 405, the 10. I think California is the only place that puts the and makes the freeways announced. So it is the 91, the 210, the 405. And we're familiar a lot with the traffic. And when I do this position as a faculty member and as the appointment as the associate dean, I have a lot of things going on. I am a clinician. I am also a researcher. I am also an educator. Sometimes I am a therapist. And so what happens is we have an intersection of a lot of things that I do. So just to give you a little bit of information about myself, I'm a native Californian, and sometimes that's a little bit interesting. I obtained my PhD from Meharry Medical College, and that's in the area of biochemistry. And then after I finished that, I went on to Vanderbilt University for my master's in nursing, where I have a dual emphasis in women's and adult health. I'm also a practicing clinician here in the LA area for a different healthcare system. Now, here in the School of Nursing, I've done a lot of things. I have been here since July 1, 2010. When you were faculty, you remember your first day that you started. And during that time, I've had the opportunity to serve on various committees here in the School of Nursing, such as the Task Force for the DNP. I was chair of that. I've been on Student Affairs. I am now currently the chair of the Curriculum Committee. That's a whole nother passion that I have. I've been on the Recruitment Committee, Faculty Committee. I'm on MAC, et cetera, et cetera, et cetera. And in the university, I've also been on other committees here as well. I am currently an equity advisor, and I'm also on the Equity, Diversity, Inclusion Council for the entire UC system. I have participated in looking at applications for students coming to UCLA, and I always take pride in this, that our students that come here are very special. The majority of our bachelor students all have above 4.0 GPA. They're sitting about 4.4, 4.5. And if I were to try to come into UCLA at this time, I would not be admitted. I enjoyed high school. I didn't do that much studying. I did take honors and AP classes, but most of our students do that. And so when you come to UCLA, it's a very leveling and humbling experience because everyone here is smart. Everyone here is gifted. Everyone here is above average. And the same thing can be said about our faculty here as well. Now, you will notice the little mice at the bottom, that you will see them come back up because at the same time of doing all of this service that I do here in the School of Nursing, as well as for the university, I have to do research. So in my clinical series, it is not clinical. In my series, my academic series, it's responsible. I am responsible for doing research, teaching, and service. And so the research component is very important for me. The research component allows me to do publications, which brings notoriety to the school as well as grant funding. It also helps us to provide opportunities for our students who would like to engage, again, shameless plug, in getting their doctorate. And one of these other responsibilities that we have is to move the science forward. And so you're balancing not only the notoriety of the School of Nursing itself, but also the notoriety of UCLA and the UC system as well. And so I will come back to my mice, who also make me very happy, and talk about a little bit of those things that are necessary in order to make sure that UCLA remains the #1 public institution nationally. Now, I'm going to pivot just a little bit because I like this quote by Shaw: "Progress is impossible without change, and those who cannot change their minds cannot change anything." And I see that this is applicable not only to my research, because it's moving the science, but also to my educational impact in the classroom. Many of my students can attest to the fact that I can be a little pushy. And being pushy, I mean that I stretch them. I have them think about things differently. I have them get into that space of discomfort because we have chosen to be a profession of clinicians that always are undergoing change. If we remember that as nursing clinicians, that we are always assessing, implementing, and then reassessing, we would understand the dynamic of change. There are some things that are going to stay pretty permanent. You are going to always like chocolate ice cream. Now, the more you make, you might start getting a little specific about what chocolate ice cream you have because you've changed that a little bit. But also you have to remember that as you go through the nursing profession, as you are clinicians, that new science comes out, new ways of doing things. You always have to remember that your patients are not always a one size fit all. And so we have to make progress in this profession, in our practice, in our research. But we also have to make sure that as we're doing that, we don't become married to so many things that keep us rigid and unable to change. And so I'm going to switch to the other part of my happy brain, which is the research. My research focus is to understand the association of IGF-2 and the development and the progression of aggressive breast cancer. And what I mean is this talk will focus on breast cancer, and I'm looking at estrogen receptor negative breast cancer, and specifically today we'll talk about triple negative breast cancer. But IGF-2 also plays a role in other gynecological cancers. And I focus on a population of premenopausal women. And the reason why this is important to me is for those of you who are advanced practice registered nurses, you will see that the guidelines for many things of women's health have not been worked out as readily and as clearly for those women who are younger than the age of 45 or 50. We take that that group of ladies for granted in that they don't have any problems. They're still considered to be of reproductive age. However, this cohort of patients will also, by and large, have the most aggressive types of cancer, especially gynecologic cancers. I'm happy that task force have changed their screening guidelines, as we'll talk about for breast cancer. But also, we are now getting more traction where clinicians are also having to talk about what is changing within a woman's body as she goes through the various stages and not just being menopausal or postmenopausal. I'm also looking in my research to assess potential use of novel oral bioavailable targeted therapies against these receptors that IGF-2 targets, and as well to see if they work better in patients that have triple negative breast cancer than those who have estrogen receptor positive breast cancer. So I get to use both sides of my brain. I get to do the basic science and molecular biology, but also I'm moving to the clinical science of also developing some novel targeted therapies. And we have several of which we are working with in the lab. I am happy to say that I actually do have a patent on several of them. And so we are looking to see how well we can get those to market. What became frustrating, though, when I learned this in graduate school is getting something to market can almost take up to 12 years. And so I'm always competing with the thought in my head when it comes to research. Am I doing enough? Am I going fast enough? Will I have time? Will I be able to make the impact? Outside of doing the publications, which is something important, I've had a paper sitting on my desk right now, and all I need to do is the discussion. And it needs to be edited and done. And then I have another paper, which I have quite a bit of data. I was looking at that to get that done as well. And so as researchers, as academics, as professors, you will see, I see students who are here and online, that we are just not in the front of the classroom teaching you. At the same time, we have the responsibilities of also doing our scholarship. And for me, that happens in the laboratory. I actually have a lab. It's upstairs on the 6th floor. It's the only one with the red door. So we have that as well. And so I am here quite frequently. So when we talk about breast cancers, we want to talk about the various subtypes that we have. Many people are familiar with those that are estrogen receptor positive. What we mean is estrogen receptor alpha positive. There are multiple receptors, and alpha is usually the designation of the receptor that was discovered first. We also have the progesterone receptor, and then we have the HER2 receptor, the human epidermal receptor number 2. And these are what we screen for by and large to see how women will respond to such medication as raloxifene, tamoxifen, how well they will respond or potentially respond to inhibitors of DNA synthesis or protein synthesis. And as you can see, these types of don't have a pointer. These types of subtypes, ER positive accounts for 75 to 70% of breast cancers. HER2 accounts for 65 to 70% of breast cancers. And because of the targeted therapies we currently have available, there is a huge response in the fact that women have the ability to live longer lives. They can have the opportunity of having low progression-free timing, or excuse me, they have a longer time before they have any progression. And some women we would consider are actually cured. Now, when you look at triple negative breast cancer down here at the bottom, what is estrogen receptor alpha negative, progesterone receptor negative and HER2 receptor negative, meaning it's not overexpressed. It accounts anywhere from 8, some places 10 to 20% of all breast cancers diagnosed annually. However, it accounts for over 50% of breast cancer mortality annually as well. And that is because we do not have targeted therapies for this type of breast cancer. And that is what I am looking at. As you can see here on our molecular signature that you would see, those that are triple negative breast cancer, from the time of diagnosis to the time of death, it drops off significantly because we do not have those therapies. And women largely that are diagnosed with triple negative breast cancer have a reoccurrence within the first 12 to 16 months. Those women tend to be African American and Latina. They also tend to be premenopausal, i.e. less than the age of 50 and many times less than the age of 45. So if we take that into context, we understand that those ladies are actually at the prime of their life. They've had their children. If they've done that, they're at their job, they're working, they're looking at a career, they have a lot of things ahead of them. The last thing they're thinking about is cancer. However, if they are diagnosed with triple negative breast cancer, these types of cancers readily metastasize. They have larger tumor size, and so we have to be aggressive with their treatment. And what we found with our research is that IGF-2 actually plays a role in blocking tumor cell death, as we see here. It is actually able to activate estrogen receptor alpha when estrogen's not even present. It is most active in cancers, and it is actually mediated in its responses because IGF-2 itself does not traverse the plasma membrane. It has to bind to receptors in order to have its actions. The other thing about IGF-2 is that it comes on and goes off many times through embryonic development. IGF-2 is required for successful placental implantation. It is required for breast bud development. It is required to be on when lactation takes place, and then it comes back on when you have breast cancer development. And so it's highly expressed in triple negative breast cancer tumors as well. We did some research looking at archival samples of TNBC tissue. And as you can see here, when we looked at the amount,
let me mute this person.
The amount of IGF-2 in normal archival tissue samples versus triple negative, you can see that there's a significant increase in our ALRED scores. Also, we found that those tumors tended to be a little bit larger in size. When we looked at other populations, we wanted to see if there was any stratification of the expression of IGF-2, and you will also see in this slide estrogen receptor beta. It's here, but we will come back to that the next time I give a presentation. We could see that IGF-2 is highly expressed in the populations of women that were African American and Hispanic, and that what you found was in African American and Hispanic women, you had a significantly higher amount of IGF-2 being expressed. When we looked at a larger cohort, because in this first cohort, we had about 80 ladies. Here, when we had a cohort of 158 archival samples, you can see that not only did you have an increase of TNBC expression in women that were African American versus Caucasian, and here you have your hazard ratio, and that being African American was actually a marker for having triple negative breast cancer. So you had more women for that did that as well. And then you can also see here that when you look at the expression of IGF-2 in normal tissue as compared to that tissue that's in tumor tissue, you actually see an increase. And we split the ladies off. In our total sample, you see that you have more IGF-2 expression in the tumors. And then we would place the ladies in early stage or advanced stage. So early stage means that you have the tumor in situ. It was smaller. It was less than 3 centimeters. You still see a higher expression of IGF-2 in the tumor. When you start looking at the ladies who are advanced tumor size, so then you mean the tumor is much higher. And as you can see, it actually went out to those women who were diagnosed with stage 4 breast cancer, which means it has already metastasized to different locations within the body. you could see that we still had more IGF-2 that was being expressed in those tumors that were triple negative. So it looks as if IGF-2 is actually a consistent marker that will bear out for the ladies significantly that have triple negative breast cancer. So it's something that we should pay attention to because we see that it's increased in early stage as well as late stage. Now, when we think about IGF-2, some of us may or may not be aware of what it does. So when you look here at this schematic, you have IGF-2, which like IGF-1, remember IGF-1 is just one because it was developed, it was identified first. And then IGF-2, they both bind to receptors as they are pictured here. It can either be a IGF-1 receptor, two of them together, or it could be an IGF-1 receptor with the insulin receptor. And what you can see here is that there are various biological effects that take place that affect cell growth, proliferation, protein synthesis, and metabolic effects, which allow for cell survival. All of the things that cancer wants to do. However, when we block the ability of these proteins, they do not traverse the plasma membrane, so they have to bind to that receptor in order for them to have their effects. But when we block their ability to bind to those receptors, in theory, we shut off all of these processes, so it doesn't go above and beyond. Remember in some slides before I showed you that there's an increase of IGF-2 that is in the tissues of triple negative breast cancer patients. And what we know is that it stimulates these biological effects. So we know for some reason the tumor has decided that this is advantageous for them to express more IGF-2. So we wanted to look at systems in which we could have a little bit more control. So this is an example of in vitro or basically in a dish. Assessment of the impact of IGF-2 on TNBC cell proliferation. And what you have here are various types of breast cancer cell lines. We created a media that was absent of every other growth hormone but IGF-2, which requires this media to be changed every 10 to 12 hours. And so in order to complete this experiment for the time frame done, we did 24, 48, and 72 hours. You can see that IGF-2 alone was able to stimulate cell proliferation by itself for a significant fold change. Now, that stimulation was not consistent across all of the breast cancer cell lines that we use, indicating that IGF-2 has differential effects based on the type of cells that are being used. So we can take away from this that also IGF-2 may have a differential effect on different types of cancers as well. Now, what we know with cancers is that usually they don't like staying in the same place. So as you can see from this schematic, you always have to have the proliferation. Then you have the ability for the cell or cells, like bad kids, they want to go somewhere and not tell you where they're going. They get into the bloodstream and they go to a very distant site. And from that site, they're like, I think we want to stay here. So then they will start to grow again and you will have different tumors. It started in the breast. The next thing you know, you might have something in the lung or you might have a tumor develop in the bladder. The primary site was the breast, but the cells have decided they want to go someplace else. And so we wanted to look at IGF-2 to see if it can facilitate TNBC cell migration. And actually we used a scratch wound test where we placed the cells on a dish. We created space so the cells would have to migrate across that space. And over 72, 48 hours, as you can see down here at the time, you can see how many days we went, and we supplemented again the media with IGF-2, which was refreshed every 12 hours to see if the cells would migrate. And it was only with IGF-2. And as you can see, if there was no IGF-2, the cells pretty much stayed on either side of the wound. However, when we added IGF-2, you could see that the cells actually moved across the wound. So we were indicating that the cells were actually able to migrate. And this is data we also published in 2019 with the team. Moving on, we wanted to say, what is the effect of IGF-2 signaling on various molecules that are responsible for migration? And as you can see here in this schematic, if you remember, receptor tyrosine kinase, that's what RTK stands for. The insulin receptor, the IGF-1 receptor, and the insulin receptor are receptor tyrosine kinase receptors. And so when IGF-2 binds to that receptor, it causes signaling to take place. And then you're able to stimulate such transcription factors such as SLUG. Now, SLUG is placed here because there's SLUG, there's snail, there's other types of mesenchymal markers or markers, as we say, that are associated with metastasis that can allow the cell to go from an epithelial phenotype in which they're happy to stay in the same place. But once you start seeing an increase of such markers as NCAD, SLUG, vimentin, and fibronectin, they're like, hey, I want to get out of here. I want to see what's out there in the wild blue yonder. So they are more readily to get into the system and go to another area. And what we have an understanding of is that SLUG, as you can see here in this schematic, actually needs to bind upstream of a promoter that's associated with BRCA2 or breast cancer receptor 2, breast cancer receptor 2. Many times we always screen for that because we know that women who have mutations in BRCA2 actually have a higher incidence of breast cancer. That mutation is associated with the development of breast cancer because BRCA1 and BRCA2 are DNA repair molecules. So whenever there's a break or damage to DNA, these proteins are released into the system, they go to the DNA in the nucleus, and they basically act as stop signals. So that there's no transcription translation that takes place. So that this DNA can then be repaired. Once it is repaired, then they come off, and then you will have healthy DNA being made. But what happens is if you have SLUG that binds to the silencer, which is 221 base pairs upstream of the promoter of the BRCA2 transcription start site, and it will then prevent the transcription of BRCA2.
Oh.
So when you have more SLUG is in binding to this inhibitory area. BRCA2 is not being made. So that allows the cell to continue to proliferate even if DNA is damaged.
Mm.
So when you think about this moving upstream for where SLUG is, if SLUG is mediated by receptor tyrosine kinase, one of which is the IGF-1 receptor, which IGF-2 binds to, then you're seeing how you have a signaling cascade that makes it advantageous for the cancer cell to continue to proliferate. And so you have more IGF-2 that's being made. So more IGF-2 binds to the receptor, sits the signaling, SLUG sits down. Therefore, we have a decrease in DNA repair. And as we can see here, when we inhibit SLUG using an siRNA, so it is not made, we turn that off, you will see more of, where's my little pointer? There it goes. You will see more of BRCA2 that is being made for the RNA. And actually, just because you have RNA does not mean that you have protein, ladies and gentlemen, because protein is what's necessary in order to mediate the effects. And so we in science have to say, okay, if you remember back in biology, you had DNA replication, then you had your messenger RNA, then you had your translation, then you had a protein, and proteins also go different modifications. So when we see more of the RNA expression, we're like, oh, that's good. More of it's being made. But now we need to make sure that that RNA sequence is being translated into a protein. And what we can see here is that when we inhibit the siRNA for SLUG, not only do we have an increase of BRCA2 RNA, we also have a decrease of SLUG protein. So we are inhibiting SLUG. And so this is helpful for us as a way of measuring the efficacy or the impact of IGF-2 onto cells, as well as a process of cells in their protein synthesis, as well as their DNA synthesis as well. Now, what I have here is that another little handy dandy twist of science. We like to see that science is either or, and that's not correct. If you remember, you have to be ready to change. That is our quote for moving forward. What is interesting is that IGF-2 actually exists in two forms. It has a pro form, which is here. It's much longer. And then it has a mature form, which is here, 67 amino acids versus 156. The cute thing about this is both of these are active within the cell. And so we are going to have an opportunity in my lab to look at which one is being favored against the different types of cell lines. As you remember, IGF-2 was able to stimulate that. But in those experiments, we used the mature form, which is the 67 amino acid. And some other data that we published back in 2011. It seems like a long time. And remember, I said it takes about 12 years for you to move anything.
Yeah.
So we're seeing that we're doing slug. The information that we did from slug was actually from my dissertation. Part of that, and some colleagues took that over. And then I was able to look at slug once again here in 2011. And so we're able to see that in estrogen receptor positive cell lines, if you use the pro form or the mature form of IGF-2, just looking to see if it increased the amount of slug being produced. MCF7s, yeah, not so much. Z7530, not so much. Mature form gave you a little bit, but not a whole lot. You see more in T47D and in Z75-1. But what's important is that these are all estrogen receptor positive cell lines. So we're seeing that IGF-2 does have an effect on this mesenchymal or metastatic marker. When you look at those that are triple negative, what you see is that the mature form of IGF-2 has an impact on the amount of slug being made for MDA231, for CRL2335, not so much for 1937, and a slight impact on HCC38. So we can see that these two cell lines here, MDA231 and CRL2335, respond readily to the mature form of IGF-2, and it makes slug, which means that we are more than likely silencing the expression of BRCA2, which means that these cells would have more DNA mistakes. But I want to take you to the salient feature right here. You also see that the pro form of IGF-2 is able to stimulate slug in the 2335 cell line. So that also highlights some differences and lets you know the unique things that may be taking place within the cell itself. Now, we also wanted to correlate this information that we saw with slug in the test tube, if you will, or in the Petri dish, along with what we saw with our archival sample, from patients. And so we stained them with IGF-2 in normal tissue or malignant tissue, and then stained for slug in normal tissue and then malignant tissue to see if there was any correlation. And what we can see again that takes place is that in normal tissue, you would see that you still have an increase of IGF-2. You're saying, well, Dr. Hamilton, that is normal tissue. That is correct. But is normal tissue adjacent to the tumor? So what there's a possibility is that IGF-2 you see more in the normal tissue, which would suggest that there's some communication taking place. We all communicate in these rooms, both verbally and non-verbally. So IGF-2 is being made more in the tumor tissue, and it's probably and likely having effect on what's going on in the tissue next to it in the tumor microenvironment. We can also see in the malignant tissue that you have more expression of IGF-2 and of slug. And then when you're looking at the expression of slug in the normal versus malignant tissue, there doesn't seem to be any statistical significance. So there's a possibility of change that is taking place here, and what is the marker, but we don't exactly know the process and what is the bigger shift. So that's something else we are looking on it in my lab. And how are we doing this? These are the experiments that are in process. We are actually doing, again, things that are taking place in the tissue culture, if you will, where we're looking at the signaling process, what communicates with what, how do they affect different processes within the cell, and the crosstalk that is taking place there. We are also looking at the tissues, the tissue culture itself. We will then isolate different types of parts of the cell, such as exosomal vesicles, along with the different if you will, compartments of the cell, dealing with the cytoplasmic, the nuclear and the membrane portion of the cell, to also look at what's going on with certain trafficking molecules that are taking place there. And then we'll do Western blot and flow cytometry to see how it changes cells, what other markers are available that we can see using flow. And then again, we have our in vivo models where we have those mice. I think you should have remembered them from the beginning. And they show up once again. These tend to be the star at the moment, because right now you can see where you have these extremely red or intense red areas. That's where we initially injected the mice and started the tumor. It's getting rather big. Yeah, mice are just wonderful, handy dandy things.
Oh.
But you can also see there's some metastasis that's here. You can start seeing the blue. And so once we injected the mice with our cells, we also give them treatment to see if we're preventing the metastasis from taking place. And you can do it multiple ways. And then once we finish that, then we isolate the tumor as well as the lungs, and we're able to do immunofluorescence, RNA sequencing, flow cytometry, and other metabolic assays so that we're able to see what's taking place within the tumor microenvironment.
What's the green stuff around the red circle?
Okay, so the intensity lets you know how many cells there are. So there's more red here. Then you're starting to see the cells in green. So there's another population of cells. And then you see the blue. So that's the furthest part that they are at. One thing we like about that, and thank you for asking that question, Mr. Espinoza, is the fact that you can get to a point where you're taking various populations of cells from the animal. You're able to know where to harvest those. And then we can compare various markers in those using RNA sequencing, immunofluorescence, for that to compare what are the potential changes. Now, I can't do this all alone. I wish I could, but I can't. It's not possible, and I don't have enough days in the time, and I got other responsibilities, as you remember from that previous slide. So this is part of the team that I work with. They're all very, very, very intelligent. One of the bonuses of working here at UCLA, I work with a colleague who is in the Department of Chemistry and Biochemistry. He developed some of the analogs, and he's also on my patent. We have to have someone who does this biostats, because that's not my thing. So you always have to have a group, and that's Dr. Ellishaw. He is amazing when it comes to numbers. And then I have colleagues who I've been working with primarily when I first started, because again, I'm an advanced practice registered nurse, but you do need to work with physicians. And so that is Dr. Richard Petrus and Dr. Diana Marquez Garban. I also need a pathologist, because remember, we were talking about what is taking place with patient samples, when I need to go and get some things from when I have permission, the proper IRB. Then I need to discuss that with a pathologist, and that's Dr. Neto Otamid. I also have other colleagues who are not pictured here who are in pathology, such as Dr. Weirdra as well. We have collaborations in that. And hopefully the next time we talk, I'll be discussing things that deal more along with the gynecological cancers in which we're looking at. Here's also additional team. I have students who have been with me before and have gone on and done great things. Gary Fernando was a Meccan student. She is now working at Ronald Reagan. Elaine Sang actually is now working on her doctorate. She is now a advanced practice registered nurse. Brittany Chow was working with me when she was here as an undergraduate student. She is currently at NYU. She is going into pathology. And Kay Foos is actually going to be a veterinarian oncologist. She is there as well. I have there's students who have gone to medical school. And then my colleagues here at UCLA School of Medicine, we have Dr. Helena Chang. She's an amazing surgeon. And then other folks who are in pathology, as well as Charles Drew University of Medicine and Science. And so those are my collaborators. I can't do it by myself. I actually still have people who are coming in now. And so at this particular time, it's 12:41. I'm happy to take any questions.
Yay! Before we do that, let me do this little thing that we like to do to keep things fun. Thank you for that, Dr. Hamilton. Awesome. So now we will go ahead and start taking questions about anything, or want to learn more about Dr. Hamilton's trajectory, her research, her work. I do have a question for you. How did you fall into this passion of yours of researching breast cancer and types of cancers?
So what actually happened was when I was in graduate school, I'm a little dating myself. Is that back at that time they did not have NIH did not have an Institute for Women's Health. They then started the Institute of Women's Health. And so they were doing research. And at the time that it came up, I was in a different lab that was working with another little interesting parasite that I had no interest in. And there was a grant that was available for breast cancer. And then my aunt had just been diagnosed with breast cancer. And so I was like, let me see what I can do with this. And so I was the lone person in the lab working with breast cancer. And so I was able to also get funding as a graduate student to continue that work. And I got to push some of the boundaries. The lab that I came out of was heavy into molecular biology. And so I took those skills and understanding those models into looking at breast cancer and looking at the cell cycle regulation of BRCA2. So that's how we came up with a repressor. And so at that time, I actually had to harvest cells every 3 hours.
Oh, wow.
So for 24 hours, I would treat them and harvest cells because I had to see how the cells went through the cell cycle. So you guys remember the G1, G2, S phase, all of that. So I did that. And that's how I kind of left it there. And then as I continued to do more reading, I was like, they don't know a whole lot of things about women. And so I continued to do research that had a focus on women and gynecological cancers. And then when I went to nursing school, I went to a school intentionally that had women's health as a specialty. Because again, women are not always cared for clinically or in the area of research as they should. And so the diversity is not always addressed. And the difference is we want to say estrogen on, estrogen off. A lot of things are happening to women who had a lot of estrogen on, so they're still in their reproductive years. So that's how I stay with breast cancer and other gynecological cancers.
Awesome.
Mhm.
Great.
Thank you for the question.
We do have a question in the chat from Mayumi. It was a great presentation. I enjoy your presentation. Do you think it is possible to develop immune therapy in the future?
Okay, so Mayumi, that's an excellent question. And actually, we're already doing that in the lab. That just wasn't part of what we were talking about today. I do have, well, now I've given myself 2 talks. I may be able to when we have done our faculty spotlight throughout the year, then we'll come back and maybe next year about this time, or it's something that I can give you an update with that because we've already done some of the research. Then that's a very forward-thinking question because you have to remember that the immune system is bathing everything. You have the cells that are going around and they're looking at, do we set up an immune response? Do we not set off immune response? Is that us or is it not? And so we have actually started doing research and answering those questions. So yes, it is possible, and that will be another place to chat about.
Cool. Awesome. We have another question from Rachel Gibbs.
Mhm.
Dr. Hamilton, do you advise we review NIH grants for possible research ideas for future PhD programs?
I would and I wouldn't. Because what you have is I have been able to sit on, oh, that wasn't on there. I sit on NIH review panels. So by and large, when you're looking at the grants that have been made available, folks are already doing the work. So it's good to know what's already being done. But it's also important to review the literature because then you're able to see the gaps. What happens, remember our quote by Shaw was to always look at the places of change. We as researchers tend to get very tunnel vision. This is what I'm doing. This is where I'm going to stay. And we forget the things to the left or to the right. And so it is a good idea to also review the literature to find out where the gaps, where people aren't looking at. And you are able to bridge from one place to another. And the reason why I say this is IGF-2, just like estrogen receptor beta that we talked about, that they were discovered secondly, but we haven't really thought about them too much. And so I am being a point of transparency is that when I go to publish things, people are like, so why is it important? It's the truth. Well, it's because it is important. And it's being shown here. And so you always have to put that up as part of your rationale because it is one of those gaps.
Great. We have another comment/question from Paul Boy in the chat. Great presentation as always, Dr. Hamilton. You briefly mentioned about the need for nursing faculty and shortage. What do you see are the reasons for this, and how is UCLA meeting the challenges with regards to obtaining new and experienced faculty to meet the needs of the students in this generation? Wow.
Now, Paul, I'm going to take a point of privilege here. He was a student of mine previously before. He himself is now in the what? The PhD program.
Yay.
So that is one of the things that we are actually doing. We are trying to engage our students, whether they're advanced practice nurses or BS students, to look at a PhD program because it answers a different question. What you're doing is you're moving forward novel science because you're looking at the gaps, what you can do. And so to answer your question, Paul, is that we are in this coming and moving forward, we are going to make it a point and intentional effort to share with our bachelor students that this is a possibility. And it's something that we really do need, and we are here to facilitate that. We are also at the process right now undergoing change to our PhD program in order so that content can be offered differently. Because unfortunately, and Paul will also tell you this, I struggle with the fact that my students actually still work while they're going to school. I think that they should be able to focus on their work because this is the only time you're going to have it. And so we're looking at opportunities to create the structure by which our students have more time in order to just focus on their work and not so much the financial pressures. Now, let's be clear, everyone. California is expensive. So if you've gone off and made California bills and then you come back to graduate school, we can't help you. You're probably still going to have to work. We are not going to pay you $3,500 a month to be in graduate school. And that's take home. That's after taxes. Just want to put that out there. So we won't be able to do that as well. But we are also looking at faculty. We are now at the point of recruitment because nursing is a clinical profession. So our faculty need to either have PhDs, they need to have DNPs, they need to be clinically experienced and or practicing. And you can be clinically practicing as an RN as well as an APRN or a PhD. So we are looking for that. That is something that we have, but it's not to say that we don't have faculty who may not be clinicians or currently practicing who can participate and provide valuable experience. We do have several faculty who are MDs already on faculty. So I hope that helps. And then I see a question, what challenges do you see to funding at the School of Nursing as cancer research versus other types of research conducted by faculty? Well, breast cancer research is very saturated. Again, that's always a challenge. And so I'm competing with larger labs, but it's always looking at the research, the current research to find the niche and to do that. You also have to publish. So that's one of the things. And so I would say a challenge, and this is my soapbox, is unlike in medicine, we have a higher teaching load for faculty in nursing than they do for medicine. It would be amazing if we could come up with a way to have either more faculty so that the teaching load for us in order to focus on research was more equitably or evenly spread across all faculty. But until we get there, we're going to have to adjust some of the ways that we do our work here in the School of Nursing.
Ooh. Another question from Cherise. School of Nursing currently recruiting junior faculty.
We will be having openings for junior faculty, so please keep your eye on our website. I know that there are some slots there on the website, and it says now we are not currently accepting positions. I discussed that with Dean Jean yesterday, and so I'll be talking to our folks here to have that updated and a little bit more inviting of where you should check back if someone's looking for a junior faculty position. So we will be doing the interviews and opening applications for those slots in the coming year. Yes.
Cool. Great.
Oh,
That's a
what is
great
it?
question.
What is a junior faculty? So what we say is a junior faculty is an assistant professor. So assistant professors are considered junior faculty. So you have junior faculty, early career faculty are considered those who are about anywhere from 3 to 6 years since they received their doctorate. Junior faculty are also considered assistant professors who are in their first 3 years.
Cool. Ariel. Dr. Hamilton, excellent presentation. A quick question. How do students learn more about diversity, equity, and inclusion at the SON?
Okay, so one thing, we have a website, and so that's on the School of Nursing website. You can click the tab that says on the banner that's Equity, Diversity, and Inclusion. We also have activities that are coming up. We will be posting the calendar of events. It will not always have the dates on it, simply because some of the activities we have will only be open to the community here at UCLA. Some might be public wide. And the other thing, I'm not exactly sure if you're asking about activities from myself as the associate dean, or about diversity, equity, inclusion in the school generally. I know that there are activities that are done in order to create inclusiveness and belonging through the student affairs unit, as well as when we are in class. There is content in which we discuss the diversity and the various populations, the struggles those populations may have during curriculum and curricular development. We also have other organizations, student organizations that are here in the School of Nursing. Men in Nursing is one that comes to mind, because men in nursing is actually an underrepresented population. And so we want to be very active. My office, with myself, I got into this appointment of October 1, 2023. So we are making a more concerted effort to work with the registered organizations here in the School of Nursing moving forward, and to help them with their development. I hope I answered the question on all points, but if not, you can let me know.
Yay. Any last minute questions? We do have only 5 minutes left. If there's anyone that has one last burning question and curiosity in the room, anyone? All good? All righty.
All right. I don't see.
That's a wrap for the first Faculty Spotlight lecture. Thank you, everyone. Woo! And join us next time later this quarter when we bring up our next faculty speaker. Have a great rest of your day. Thank you.
Bye, everyone. Take good care.
That was great.
Thank you.
Now my head is swimming. Trying to figure.
Is it?
Yeah, I was like way over my head, but that's okay.
Well, I tried.